Hey everyone, I Komal Dhapola from MSc Microbiology 2nd SEM is going to deliver a presentation on prokaryotic transcription. Please like, share and subscribe and don't forget to press the bell icon. Now, transcription. The process of synthesis of RNA by copying the template strand of DNA is called transcription. During replication, the entire genome is copied, but in transcription only the selected portion of genome is copied. The enzyme involved in transcription is RNA polymerase. Unlike DNA polymerase, it can initiate transcription by itself. It does not require primase. Now, steps of prokaryotic transcription. The mechanism of transcription completes in three major steps. First one is initiation, second one is elongation, and at last termination. In initiation, firstly, the transcription begins when the DNA-directed RNA polymerase associates with the sigma factor to produce holoenzyme. Basically, the holoenzyme, it is required for correct initiation of transcription binding to promoter. Then the sigma factor allows the polymerase to bind specifically at the gene's promoter sequence. There are two promoter regions present. They are Pribnobox and -35 sequence. Here we can see in this diagram, this is the -10 region which is known as Pribnobox, and this is -35 region. Basically, in Pribnobox, which is -10 region, it is found in 5 to 10 bases to the left or upstream from the first base, which is copied into mRNA. It contains a hexamerase consonant sequence which is TA-AT, of which the T at 6th position called conserved T because it is present in almost all prokaryotic promoters, analyzed by 96%. Now in 35 sequence, it is present in 16 to 19 bases upstream from the Pribnobox. It also contains a hexamerase consonant sequence which is TT-GACG. This region is the initial binding site for the sigma subunit of RNA polymerase. Now, in this diagram, we can see that this is the transcription initiation by prokaryotic RNA polymerase. How the initiation works in prokaryotic transcription. Basically, firstly, the first stage of transcription is the formation of an open promoter complex. Firstly, the sigma subunit of the RNA polymerase recognize and bind to -35 sequence, which is followed by the binding of core enzymes. Then the RNA polymerase, by virtue of its large size spans region of 17 to 19 base pairs and gets bound to the Pribnobox, this early stage structure of binding of RNA polymerase holoenzyme to the -10 region is called the -35 is called the -10 region is called closed promoter complex. Here we can see this this is the closed complex. Then this RNA polymerase in turn causes localized melting. Here we can see this is the localized melting, that is unwinding of DNA helix. This conformation stage is called open promoter complex. Then the unwinding spans in a region of about 10 base pairs from left end of the Pribnobox and extending about 20 base pairs after the position of the first transcribed base. Then the melting is necessary for the pairing of incoming ribonucleotides. After then, here we can see this is the open complex. Then this after open promoter complex formed, then the polymerization started. Then the RNA polymerase contains two nucleotides binding site called the initiation and the elongation site, which is also known as catalytic site. Now, the initiation site primarily bind purine trinucleates ATP or GTP. Therefore, the first nucleotide is to be incorporated is ATP or GTP. Thus, the first DNA base transcribed is either thymine or cytosine. Then the initiating nucleoside triphosphate binds to the enzyme in the open promoter complex and forms a hydrogen bond to the next base in the DNA strand. Then the two nucleotides are then joined together. Once RNA polymerase moves on the DNA behind the enzyme closes as the hydrogen bonds of the DNA base pair reforms, Then the enzyme reads the template strand in a 3' to 5' direction, as it synthesizes mRNA in a 5' to 3' direction. Now in elongation, after several nucleotides are added to the growing chain, the RNA polymerase undergoes a conformational change and the sigma subunit dissociates. Then the strands of double-stranded nucleic acids, even in temporary hybrids such as DNA, RNA molecules must be antiparallel if hydrogen bonding across the strand is to take place. Then the energy required for synthesis is provided by the triphosphates ribonucleosides. These ribonucleotides are the energy sources, which is building blocks, and the information components for mRNA synthesis. Here we can see in elongation in prokaryotes. During elongation, the prokaryotic RNA polymerase track along the DNA template synthesis mRNA in the 5' to 3' direction and unwinds and rewinds the DNA as it reads. Here we can see this is the 5' to 3' direction. This is the synthesis of nucleotides, and this is non-template strand, and this is template strand. Now in termination, there are two mechanisms of termination. First one is row independent. Basically in this mechanism, transcription is terminated due to specific sequence in terminator DNA. The terminator DNA converts invert repeat, which causes complementary pairing as transcript RNA form hairpin structure. Then here we can see in this diagram, this is the row independent termination. This is the template. This is the DNA double DNA strand, and this is the direction 5' to 3' and 3' to 5'. Basically in this, the RNA polymerase slow down and pauses due to the formation of hair loop structure. Here we can see this is the formation of hair loop structure, and after the continuous string of U nucleotides destabilize of RNA polymerase, then RNA polymerase dissociate and transcription is terminated. Now in row dependent termination, in this mechanism, transcription is terminated by row protein. Basically it is a ring shaped single strand binding ATPase protein. The row protein binds the single stranded RNA as it exits from RNA polymerase enzyme complex and hydrolyze the RNA from enzyme complex. Basically row dependent termination we are using when the row protein, the process is known as the row dependent of transcription. This is the diagram of row dependent transcription termination in prokaryotic. Firstly, the RNA polymerase transcribes DNA. Then row factor, this is the row factor attached to the recognition site on the RNA. Then after that, this row factor along the RNA following RNA polymerase. This is the RNA polymerase. This requires energy. Then after that, this row unwinds the DNA RNA hybrid in the transcription bubble. Here we can see this. Then after that, transcription is terminated. Then RNA polymerase, row factor and the RNA strand are released. Thank you.